Very good afternoon to one and all. The next topic is mean absorption time. The difference between the MRT after oral administration and after IV administration. So here in this mean absorption time, you are going to calculate the mean residence time for two different routes, that is oral as well as through IV. So this is called the mean absorption time, representing the average time required for the drug to reach the systemic circulation from the GIT. So when you take the dosage form through oral administration, after reaching into systemic circulation from the GIT tract, you need to calculate the mean absorption time by using these equations. Mean absorption time is MRT oral minus MRT IV. Mean residence time of oral administration subtracted with mean residence time of IV administration. So you want to know MRT for IV, then you have to use our equation, that is AUMC by AUC. So this is the normal equation for calculating MRT, right? So in this case, when you consider IV administration, you have to calculate this MRT by using this area under the first movement curve, area under the curve. And in case of MRT for oral, the equation is area under the first movement curve of oral administration divides with area under the curve with oral administration. So these are the equations for calculating the mean absorption time. So in case of extravascular and in case of IV administration, so what are the parameters you need to consider here? So this A compartment, you can assume it as central compartment. And then this one compartment is peripheral compartment. So when your drug is transported from central to the peripheral, in case of extravascular administration, here there is a pharmacokinetic parameter you have to calculate, must and should. This is absorption rate constant. Ka is absorption rate constant. This will be elucidated out in case of extravascular administration. So in this extravascular administration, your drug will be transported from the central compartment to the peripheral compartment. Here, there is a chance to get fraction of bioavailability about 200%. And in case of intravenous infusion, so the absorption will take place from peripheral compartment. Then here it is having some elimination rate vascular administration, and this is the intravenous infusions, right? So you want to calculate the MRT for extravascular administration, you have to take AUMC divides with AUC. This will gives you MRT for system compartment A plus 1. In case of extravascular, your drug is transported from the central to the peripheral compartment. So there is a need to combine these two MRTs. You will get an extravascular mean absorption time. Then you want to get MRT for this IV infusion, you have to use this equation. Area under first movement curve divides with area under the curve of intravenous infusion. And then MRT for compartment A. So MRT for central compartment. Compartment A in the sense that is central compartment. So you have to get this MRT for this central compartment, that is compartment A. You need to subtract it with this extravascular MRT and then intravenous MRT. So that is MRT compartment A is equals to MAT, mean absorption time. This is mean residence time. So because you will get bioavailability 100% here in case of extravascular administration. And then this mean absorption time is calculated from the plasma data, is the MRT at the injection site. In case of this MRT, your fraction of bioavailability is 100%. So otherwise you can calculate the mean absorption time is equals to mean residence time of compartment A, that is central compartment, divides with the fraction of bioavailability. Then the next parameter is mean dissolution time. So what do you mean by mean dissolution time? In case of tablet, in case of solid dosage form, when you take into consideration of this tablet dosage form, it will undergo dissolution. After undergo this dissolution process, it leads to the formation of a solution. So the solution is available for your drug absorption. So the drug which is available in the form of solution, that will be useful for doing its absorption. So this will be available in the digestive tract, and this absorbed solution will be available in the blood circulation. That is nothing but your systemic circulation. So in this phenomenon, you need to calculate the mean dissolution time of a dosage form. That is, mean dissolution time of a dosage form is mean residence time of a tablet minus with mean residence time of a solution. That means when you take solid dosage form into consideration, here you may observe two different phenomena. First one is your tablet is available as a solid dosage form, as a solid. Your tablet is available as a solid in initial stages. When it will be go in contact with the solution, it will be available in the form of solution. So that's why you need to calculate the time, mean residence time, in the form of solid form and in the form of solution. So when you subtract this solution form and the solid tablet form, you will get the mean dissolution time. These mean absorption time and the mean dissolution time are the phenomenons which explains you the mean residence time for different compartment models. So with that, I completed second topic of 7th unit. So the third one is physiological pharmacokinetic model. This is one of the important non-compartmental analysis of pharmacokinetics through the physiological models. So physiologic pharmacokinetic models is also called as blood flow rate limited model or perfusion or membrane rate limited models. So here this model will be assumed depending upon the anatomical and physiological data, okay, of the animal or of the human beings. So it is depending upon the designing of experiment. The number of compartments to be included in this model will be depend upon the drug deposition characteristics. So we cannot say that here lung, kidney, okay, and all the parts, heart, brain, all the organs will be involved. We cannot say that. So it will be totally dependent upon the drug deposition characteristics. Deposition will includes distribution, metabolism, and excretion. So based on these three mechanical properties of the drug, your compartments may be designed, drawn on the basis of anatomic and physiological data. Organs or tissues having no perfusion are excluded in this model. So which are showing this no perfusion? Those are of bones. Bones doesn't shows any perfusion, so you have to exclude the bones in this case physiologic pharmacokinetic model. So the drug movement to a particular region is much more rapid than its rate of drug delivery. So your drug is having movement, it is more rapid than the rate of drug delivery. So your drug is transported between the various organs is very fastly, and then it is said to be perfusion rate limited. Thus, it is applicable to highly membrane permeable drugs. So, this physiologic pharmacokinetic model can be applied to highly membrane permeable drugs. That is, low molecular weight, poorly ionized, and highly lipophilic drugs can promote the physiological pharmacokinetic model. For highly polar, ionized, and charged drugs, this model can be referred as the membrane permeation limited. So, if your drug wants to distribute or metabolized or excreted between the organs or tissues which are totally dependent upon the anatomic and physiological data, you need to analyze some pharmacokinetic parameters. So you have to take consideration into the molecular weight, the lipophilicity, the nature of the drug, either it may be lipophilic or hydrophilic. Okay, so here two cases you have to consider. First case is highly membrane permeable drugs, and you have membrane permeation rate limited. So in case of highly lipophilic drugs, okay, these drugs will be traveled through this highly membrane permeable, because these lipophilic drugs are having some solubility, lipid solubility. So your membranes are also made up of with lipids. So in the composition of cell membrane, we already studied in the first unit. In the composition of cell membrane, it is having lipids. So because of the lipids, these are prone to have some permeation in case of this highly membrane permeable. And in case of membrane permeation rate limited, the drugs which are showing charge, or the drugs which are having polar in nature, or those are ionizable, those are said to be permeation rate limited. And then what are the merits and demerits through this physiological model? Ideally, it will provide an exact description of the time course. So it is telling us the time course of the drug exactly. For example, when you take liver, then kidney, then lungs, this is lungs and this is liver, how much time it will be spent on individual organ, you can calculate very easily. Based on the, what parameters you can calculate? Based on the volume of the tissue or organ, volume, and then based on the anatomical structure, that means the surface area provided by the organ or tissue. So the surface area is also very important parameter to distribute or to deposit the drug, right? So the parameters of these models will correspond to actual physiological and anatomical measures. And with this method, we have some demerits also. Greater requirements for in vitro or in vivo data. When you want to do the experiment in the lab scale or in the animal models, you have to require some great requirements. Statistical evaluation of this model variability will have challenge. Model dependent and implementation requires appropriate expertise. So we cannot calculate the pharmacokinetics very easily as compared with one compartment open model. So this is somewhat complicated process. That is, it is totally dependent upon the distribution and the size of the organ and volume of the organ. So there are number of parameters will influence this model. So what are the assumptions which are useful to do this pharmacokinetic calculation here? What are the assumptions? There is an exchange of drug between the capillary blood and the interstitial water. So the drug will be exchanging between the capillary blood and interstitial water. It is considered to be very rapid, and the cell membrane is considered to be very permeable to the drugs. Capillary membrane does not offer any resistance to drug permeation. That means this capillary membrane is having permeable nature. It doesn't have any resistance power to the drug permeation. And there is a constant ratio of drug concentration between the organ and venous blood. So always you need to estimate here the concentration of the drug between the artery and veins. This model can be drug movement and deposition in the body based on the organ blood flow and the organ spaces. Spaces are nothing but the surface area provided by the organ. So how the drug is moved here? The drug movement and the deposition in the body is depending upon the blood flow to the organ and the surface area provided by the organ. It is the simplest form. A physiologic pharmacokinetic model considers the drug to the blood flow limited. Drugs are carriers to organs by arterial blood and leave organs by the venous blood. Everyone knows this. The drugs will be carried out by the arterial blood and then it will be leaved out from the venous blood. So you need to calculate here the pharmacokinetic parameters on the base of mass balance equations, which are written for each compartment model to describe the inflow, outflow, accumulation, and the disappearance of the drug. So how you need to calculate the pharmacokinetic parameters using this model. So in this model, the blood will be carried out through the arteries and it will be eliminated out with the venous blood. So you need to measure here QT. QT is nothing but the rate of blood flow. The rate of blood flow. So here you have to measure always the rate of blood flow. That is nothing but the QT. CRT is means arterial drug concentration. C stands for concentration, ART stands for artery. Here C stands for concentration, VEN stands for vein. So when your drug will be entered into the tissue compartment or blood, it will be carried out by this C vein. Okay. So uptake of drug into the tissues is very rapid and a constant ratio of drug concentrations will be travel between the organs and the venous blood. See, this is the organ and this is the venous blood. So there is a distribution of your drug will be very rapidly. And then you need to estimate here the partition coefficient. Why? Here we are observing that our drug is distributed between two different compartments. You just assume like that. Okay, one is tissue compartment and the other one is blood compartment. So you have to estimate the partition coefficient ratio, that is tissue blood partition coefficient, that is P of tissue is equals C of tissue divided by C of blood, that is concentration of the drug in the tissue divides with concentration of drug in the blood. So this will states you about the partition coefficient of the drug between the tissue and the blood. So this is of equation 1. P is the partition coefficient here. And the most important parameter here is the partition coefficient can be varied on the type of the tissue. So in case of adipose tissues, this is consisting of majority of fats, right? So in these adipose tissues, your hydrophilic drug is having very less partition coefficient because the drug will be solubilized with aqueous medium. Those are of hydrophilic molecules. If your drug is having lipophilic in nature, so it will be solubilized in lipid contents, right? So these adipose tissues are having fats. So that's why this lipophilic drugs will shows a high partition coefficient in that. So the partition coefficient can also be varied on the type of the tissues. So in case of other tissues, your lipophilic drugs may not get that much high partition coefficient. The only reason is the adipose tissues will have some fats. These are useful to get a high partition coefficient in case of lipophilic drugs. In case of hydrophilic drugs, in the same adipose tissue, your drug is having low partition coefficient. That is the difference. And then the rate of blood flow to the tissue can be expressed as QT. This is rate of blood flow to your tissue or organ. So the rate will be ml per minute. So this is will says you the volume and this will says you the time. So that is what minutes, that is what the time. So how much volume of your blood will be transported to the organ in what time, how much time that will be. So the rate of blood flow to the tissue will be expressed you QT, and the rate of change in the drug concentration with respect to the time. Here your drug will be changing its concentration with respect to the time within a given organ or tissue, which can be expressed as, see here, the equation is previously what we studied, the equations of dC by dt. What do you mean by dC by dt? This is the rate of process, right? This is the rate of drug process. So here instead of writing this C, you can write V of tissue, C of tissue. Why means here your drug is transported between the blood and tissues. Okay, so that's what you can write instead of C here, V of tissue and C of tissue. Then divides with dt. With respect to the time, your concentration of drug will be varied. Okay, this is gives you the QT, that is rate of blood flow. Rate of blood flow with of C in minus C out. That is the concentration of the drug in and the concentration of the drug out. So in is nothing but your artery. So the concentration of the drug will be taken up by the artery and it is leaving up by the vein. So that's why the out is nothing but the vein and in is nothing but your artery. So your equations will be replaced with in and out with artery and vein. These are the very simple equations. Okay, once you go through it, it is very easy to learn. Where C art is the arterial blood concentration, C vein is the venous blood concentration, QT is the blood flow, and it is represents the volume of blood flowing through the typical tissue or organ per unit time. Here V stands for volume of the tissue. C stands for concentration of the tissue. So very simple. Okay. So if the drug uptake occurs, in the tissue, the incoming concentration will be CRT, which will be higher than the outgoing venous concentration. What is the meaning of this sentence exactly? If your drug will be uptaking within the tissue, so here you have a tissue or you have a blood, right? So the drug will be uptaken by this tissue. Definitely there is a artery concentration is more than that of vein concentration. So in the blood flow limited model, the drug concentration in the blood leaving the tissue and the drug concentration within the tissue are equilibrium. So with respect to the blood flow limited model, your drug concentration will be same, equilibrium in the tissues and the blood. So when the blood flow is same to the tissue in the blood, here the concentration of the tissue will be, the concentration of the drug available in the tissues are equilibrium. So then your C vein may be estimated from this equation, that is D of volume of tissue, concentration of tissue, concentration of drug in tissue divides with dt is equals rate of blood flow into C artery minus C tissue by P tissue. So in case of blood flow limited model, the drug concentration in the blood leaving, that is nothing but C vein, and the concentration within the tissue, the concentration which is having the tissue concentration, both are having equilibrium. And then this equation will describes you the distribution of a non-eliminating organ or tissue group. For example, drug distribution to the muscle or adipose tissue and the skin, which is represented in a similar manner. In that cases, here there is a negligence of your venous blood concentration. In case of skin, adipose, muscle, so these all organs will consisting of you some fats. So these will restrict the blood flow. So this is the blood flow to the organs in this physiological model. So you already know about capital Q. Capital Q represents you the blood flow. Just remember this, blood flow to organ. So see here this is the venous blood and here this is the arterial blood. So arterial blood is taking the drug and then venous blood is leaving it, right? So then here you have different kinds of organs or tissues. See here, brain, lung, heart, muscle, kidney. And then here you have a liver. From this liver, the distribution of drug is happen in two different kinds of organs. What are those? Spleen and GI tract. And here you have bones and adipose tissue and skin. So if you want to calculate the blood flow to the particular organ, you need to have a denotation, right? That is Q of BR. That is blood flow of brain. This is blood flow of lung. This is blood flow of heart. So that's why Q called as blood flow to the particular organ. So only one thing you have to remember, whenever you need to calculate the blood flow to the liver, you have to take consideration of the spleen and GI tract, because from the liver the drug is transported to the spleen and GIT. So that is what important point. See here, from this upper body, see, this is the right heart and this is the left heart, and this is what, the lung. So from the liver, here your drug is deposited in the spleen or intestine. So this is the left heart, this is the kidney, and this is the lower body. So wherever you need to calculate this Q for this liver, you need to take QS plus QI. QI nothing but QGIT. The physiologic pharmacokinetic model assumes flow-limited drug distribution without drug binding. So here it is taking consideration only the flow, not to the drug binding. Your drug may be bind to either of plasma proteins or any ligands. Okay, this is of without drug binding. Only it is taking consideration of flow. In reality, many drugs are bound to variable extent in either plasma or tissues. With most physiological models, drug binding is assumed to be linear. So this physiological model will be applicable. It is taking the consideration into the flow, okay, not the drug binding. But in general terms, many of the drugs will be bound to the various types of plasma proteins, right, or ligands. So if in that cases the drug binding should be linear, you can assume like that. So bound and free drug in both tissue and plasma are in equilibrium. It is having tissue and plasma equilibrium. Further, the free drug in the plasma and the tissue equilibrates rapidly. So you have a bound drug and you have a free drug. So both will get an equilibrium in the tissues and in the plasma. So the free drug concentration in the tissues and the free drug concentration in the emerging blood are equal. So the equations will be like this. So C of B is nothing but the total drug concentration in the blood. So that is equal to C of T. This is the total drug concentration in the tissue. Here small t stands for tissue and small b stands for blood. So here C of T is concentration of drug in tissue, C of B is concentration of drug in the blood. So what do you mean by the F? F is F of B. Small f stands for fraction. Fraction of the fraction. And then F of B, see here, F of B is the blood free drug fraction. Blood free drug, that is unbound drug fraction. F of B, see here, F of B, that is blood free drug fraction, that is unbound drug. Free drug means unbound drug. So when you calculate the C of B, you have to calculate the normal fraction. Then it will be, the fraction will be in the sense of this blood free, that is unbound drug fraction. You will get the equation like this. This is the unbound drug fraction, and Ft is the tissue free drug fraction. That is, tissue free means in these tissues your unbounded drug will be available. So you need to calculate the free drug for the blood and the free drug for the tissues by using these two equations. And this is the generalized equation for calculating the fraction of the drug which is available in the blood and in the tissues. So here you need to calculate the partition coefficient ratio, that is PT, partition coefficient ratio of the tissue drug concentration to that of tissue plasma drug concentration. That is F of B. F of B is blood free drug fraction divides with F of T, that is tissue free drug fraction, with is equals concentration of drug in tissues, concentration of drug in blood. That will gives you the partition ratio, that is of PT. And then by assuming the linear drug binding, so I already told you few drugs will be bound to either plasma proteins or blood proteins, right? So in that cases the binding should be linear. So there we will get a rapid equilibrium. The free drug fraction in the tissue and blood may be incorporated into the partition ratio. So for that reason we need to calculate the partitioning of the drug concentration in the tissue and blood. Okay, so the equations are similar to the above. The drugs which are having free drug concentrations are substituted with the CB. Drug clearance in the liver can be assumed with free drug only. The inherent capacity for drug metabolism will be denoted you as CL int, that is inherent mechanism. See, general mass balance equation for various tissues. So here you have to remember the mass balance equations for different tissues. Okay, this is the basic equation. And then if it is a liver metabolism, so I already told you in case of liver you need to remember the spleen and GIT. Why? Because here the artery is involving the drug inside, and then here the vein is involving to leave the drug outside. So for that reason, whenever you want to calculate the liver metabolism, you need to calculate GI and SP. SP is nothing but the spleen. See, this is our basic equation, no? You have to substitute your liver here. See, D of volume of liver divide into multiplied with concentration of liver divides with dt. With respect to the time, the volume and the concentration of the organ will involves. Then C of B of Q of liver minus Q of GI and Q of SP. Q is nothing but blood flow. Blood flow to the GIT, blood flow to the spleen, and blood flow to the liver. Then you need to get this hepatic drug elimination. So these are the general mass balance equations for the drugs. For the organs or tissues, you have to calculate the pharmacokinetic parameter here. So in the same manner, if you want to calculate this general mass balance equation for brain, for heart, instead of writing this liver, you have to write brain and heart likewise. Okay, so you have to calculate so on equations. Mass balance for the drug in the blood pool, it is total blood pool. See here how the equation will be. This is the volume of blood, this is the concentration of the drug in the blood divides with dt. See, Q of MUS, MUS is nothing but muscle, and LIV liver, KID kidney, skin, and then fat adipose tissues, and LU lungs, and then finally it is a blood. So this physiological pharmacokinetic model is very simplest model. Here we are assuming that our drug which is intended for oral use can be deposited between the compartments like our organs or tissues. So only one thing you have to remember here, we are considering our organs or tissues. The blood will be the organs or tissues. The drug deposition will be depending upon the volume and the blood flow to the organ. So the drug is depositing on these two parameters. You are going to calculate the Q for all the organs separately. Then you have to know the mass balance equation for the blood pool. You need to add all these Q's. Okay, so the influence of binding on drug distribution is an important factor in the pharmacokinetics. Only the animal data may predict the drug distribution in the humans. So how you will differentiate the blood flow limited model and the diffusion limited model? In case of blood flow limited model, there is a rapid equilibrium assumes. So here you are getting some rapid equilibrium. That is, the drug diffusion is extremely fast and that the cell membrane offers no barrier to drug permeation. In case of blood flow limited model, there is no need of requiring any barriers to drug permeation. Directly, the drugs will have highly permeable to the blood flow limited model. So there is a need to get rapid equilibrium. So in case of diffusion model, here cell membrane act as a barrier. So with the top cell membrane, your drugs will get diffused into. So once you remember that cell membrane structure, okay, so then it is easy to understand the concept. And in case of blood flow limited model, if no drug binding is involved, the tissue concentration is the same as that of venous blood. That is the living tissue. Blood flow is very rapid and drug permeation is slow. A drug concentration gradient will be developed. See, you just remember one thing. Whenever your drug molecules will be diffused by the use of cell membrane, there it will develop some concentration gradient across the cell membrane. With the top concentration gradient, your drug molecules will enter into the systemic circulation. What we learned the concept under the absorption topic. So here, this is the rate limiting step is blood flow, and here the rate limiting step is permeation. And this assumption greatly simplifies the mathematics involved, and this is the time lag equilibrium between the blood and tissues. So in case of this diffusion model, here we are estimating the time lag between the blood and tissues. So by this way, you can differentiate the pharmacokinetic models for diffusion model and blood flow limited model. So here you have some examples of the drugs which are undergo the physiological pharmacokinetic models. So in case of thiopental, it is acting as an anesthetic. So here it is going the physiological model through blood flow limited. And in case of cisplatin is an anti-neoplastic drug. It is undergo this plasma multiple metabolite binding. So nicotine, lidocaine, methotrexate and piperidine, these all drugs will follow this blood flow limited model, that is a physiological model. Okay, only this cisplatin is undergo this plasma multiple metabolite binding. So here this is a very simple mechanism for explaining the non-compartmental analysis. So in case of this non-compartmental analysis, here we are not assuming that any compartment is there for drug distribution, right? So in this physiological model, what is our assumption? Our drug will be transported between the various organs or tissues. So drug is transported is nothing but drug is deposited. So in depositing, you have to take consideration into distribution, metabolism and excretion. So the drug deposition characteristics can be estimated through all these organs or tissues which are present in our body when you take the drug either oral or transdermal or whatever it is. So it is totally dependent upon the blood flow to the organ, and one more thing, the volume of the organ. So your drug deposition can be dependent upon the blood flow to the organ and the volume of organ. From that you can calculate the rate of blood flow to the organ. So this is what the simplest model of physiological pharmacokinetic model. With this, your 7th unit is completed.